chromosomes, which I have placed after chromosome 7, as befits the X chromosome size. You now know why the last chapter of a book that boasts in its subtitle that it has 23 chapters is called Chapter 22. It is, at first glance, a most misleading thing that I have done. I may seem to be implying that chromosome 1 came first, which it did not. I may seem to imply that chromosome 11 is exclusively concerned with human personality, which it is not. There are probably 60,000 to 80,000 genes in the human genome, and I could not tell you about all of them, partly because fewer than 8,000 have been found, though the number is growing by several hundred a month, and partly because the great majority of them are tedious biochemical middle managers. But what I can give you is a coherent glimpse of the whole, a whistle-stop tour of some of the more interesting sites in the genome and what they tell us about ourselves. For we, this lucky generation, will be the first to read the book that is the genome. Being able to read the genome will tell us more about our origins, our evolution, our nature and our minds than all the efforts of science to date. It will revolutionize anthropology, psychology, medicine, paleontology, and virtually every other science. This is not to claim that everything is in the genes, or that genes matter more than other factors. Clearly they do not. But they matter, that is for sure. This is not a book about the Human Genome Project, about mapping and sequencing techniques, but a book about what that project has found. Sometime in the year 2000, we shall probably have a rough first draft of the complete human genome. In just a few short years, we will have moved from knowing almost nothing about our genes to knowing everything. I genuinely believe that we are living through the greatest intellectual moment in history, bar none. Some may protest that the human being is more than his genes. I do not deny it. There is much, much more to each of us than a genetic code. But until now, human genes were an almost complete mystery. We will be the first generation to penetrate that mystery. We stand on the brink of great new answers, but even more of great new questions. This is what I have tried to convey in this book. Primer. The second part of this preface is intended as a brief primer, a sort of narrative glossary on the subject of genes and how they work. I hope that readers will glance through it at the outset and return to it at intervals if they come across technical terms that are not explained. Modern genetics is a formidable thicket of jargon. I have tried hard to use the bare minimum of technical terms in this book, but some are unavoidable. The human body contains approximately 100 trillion million cells, most of which are less than 1/10 of a millimetre across. Inside each cell there is a black blob called a nucleus. Inside the nucleus are 2 complete sets of the human genome, except in egg cells and sperm cells, which have 1 copy each, and red blood cells which have none. 1 set of the genome came from the mother, and 1 from the father. In principle, each set includes the same 60,000 to 80,000 genes on the same 23 chromosomes. In practice, there are often small and subtle differences between the paternal and maternal versions of each gene. Differences that account for blue eyes or brown, for example. When we breed, we pass on 1 complete set, but only after swapping bits of the paternal and maternal chromosomes in a procedure known as recombination. Imagine that the genome is a book. There are 23 chapters,